Okay, why I thought I was happy to volunteer for this presentation was because most of my um, academic studies were also done part-time whilst working. So I've got a fair story to tell with over 35 years experience um, in drug discovery. So I'm a group leader at Charles River, as it's been said already, and I've worked as both biochemist and analytical scientist for those 35 years. I have experience in analytical biochemistry, cell biology, pathology, and drug metabolism, and I've worked on a large variety of therapeutic areas. I obtained both my degree and master's whilst working for Rome Prolong. So here's my career journey. I started straight from school as a student radiographer in 1982, and I studied. I practiced at Chelmsford and Essex Hospital, and but the studied had a study day every day a week at Barts, and actually this was an NHS grant, and I was only a student for 3 years rather than being paid. And then at the end of that 3 years, you had to apply for jobs as radiographers. And after a temporary job at St Mary's in Paddington, I ended up at St Bart's Hospital. And worked there for 18 months. Whilst it was quite enjoyable as radiographer, two things happened that I hadn't appreciated as a student. People became parts of bodies, and someone would say there's a trolley for a chest in the corridor and all of that sort of thing. So that wasn't very pleasant at times, and it was quite a busy, productive sort of, it was like almost like a process pushing people through to have their x-rays, and also in those days anyway, you worked really long times because you had worked your day and then you worked overtime, and it's just a long drag. And so I was looking for something more local, couldn't find it, but a personal advert appeared in the local papers, advertising for junior technicians at what was May and Baker at Dagenham, which was nice and local. And as I was newly married, I thought that was the other type of profession I'd looked at when I chose to do radiography. So I took a bit of a gamble and quite a pay cut, and moved into Dagenham May and Bakers as a laboratory technician in 1987, and enjoyed it from day one basically, and moved through the ranks quite quickly. They offered part-time education. As I had A levels, I was able to do what the BTEC Higher National Certificates, which was a 2-year part-time course in applied biology. And if you wanted to, that then led to a 3-year part-time degree, which is very similar to the apprenticeships we have now through University of Kent, where you do 3 years for a foundation degree and 2 years for the full honours degree. So whilst working at Dagenham, initially I started in vascular research, doing cell culture, smooth muscle cells, and we were looking at fatty acid metabolism, and we isolated various things by chromatography. These cells were fed with radioactive carbon-14C, and that meant that they could convert this into fatty acids, which we then isolated by chromatography and read on radio scanners. So that was my job for 18 months, and also preparing media, various things as well. Whilst I was enjoying that, an opportunity came to move into the pathology group, and in the pathology group, that seemed to be more akin to what I've been doing in the hospital as well. So I moved in as a clinical chemist, or trainee clinical chemist in pathology at Rome Prolong. It was a very small group, there was only 2 clinical chemists, and I was one of them, a histologist and a hematologist. And so I definitely got to do an awful lot, and I really took to clinical chemistry. Did a second HNC in a year for that, and never looked back really. So moving on to the next slide, if I can. From 1987 to 1989 to 1997, I was in pathology as a technician, and then scientist, senior scientist, and right up to research scientist, where I was analysing plasma samples from both humans and animals, and looking at different organ function profiles, various hormonal measurements, a huge number of different techniques, from radioimmunoassay binding, ELISAs. I was actually the first person at Rome Prolonged to do in our biology department to set up an ELISA, electrophoresis for various lipid metabolisms, flow cytometry towards the end of period in pathology, and basically really enjoyed being able to look at disease profiles and animal models, and compare the profiling across a number of different projects. Whilst there, then I got my bachelor's from Stratford, University of East London, and then also because I then was going to conferences and various clinical chemistry topics and immunoassay topics. I went to I think the very first ELISA Congress, which was set up in Florence in Italy, and at that conference there was some discussion about how you could take some genetic information and look at prognosis of disease in patients, which tempted me to then explore this master's, which was molecular pathology. And whilst I was doing that, my thesis was on procollagen expression in lung fibrosis. So moving on from there. Whilst I was in pathology, whilst doing a large amount of clinical chemistry, we also were able to collaborate with universities across London, because we're so close, and we established within the pathology group, we established a lung fibrosis model and screening, looking at collagen deposition. So it was at a cellular level, and then we were also later on, as this became more of a project across the whole of biology, we were looking at histological samples of lung fibrosis. Also, I had a large amount of bone research, so we were looking at how, when we had an asthma project, how treatment of steroids affected other circulatory and organ systems. And so my part in that was looking at bone growth templates and osteocalcin deposition to look at bone, which went akin to what I've been doing on my degree, which alkaline phosphatase isoenzymes for bone changes there. It was a very interesting type of work, and I think whilst I was in this group, the most interesting thing we ever did was a collaboration with Guy's Hospital, where we were looking at patient x-ray patterns and comparing them to macroradiography of mouse joints, and looking at biochemical changes there between and trying to compare the radioactive changes to the biochemical plasma changes. So that work was ongoing when, unfortunately, in 2000, the Dagenham Research Centre was closed. But prior to that happening, I was asked to, the clinical chemistry work dried up, so I was asked to actually take on lead for a group of histologists. Whilst I wasn't a histologist, I was able to guide them in how to control assays and set up immunohistochemistry, and then we also moved into in situ hybridization in tissues. And I led all of that work at the time. By now a senior research scientist. and also then went to train with the veterinary college in Edinburgh to read slides, certainly just on lung histology. So, but that was a very interesting last 2 years of my time at Dagenham Research. Whilst it was announced at the end of 1999 that we were going to be made redundant as they closed the whole research site. Unfortunately, that period, I also found out I was having a baby. So my redundancy time was spent basically on maternity leave, and that did enable me to have a slightly longer period, but also meant that I wasn't really sure whether I could, what work I might be doing once I'd had the baby. I went for a few interviews, but really wanted to just see how the land lied by the time I was ready to go back to work. And then when I was ready to go back to work in 2002, or at the end of 2001, I was approached by one of the scientists I'd worked with at Rome Rora, as it had become by the time we left. And in 2000, a group of scientists from Rome Rora decided to set up a spin-off company, which was called Argenta. At the time, Rome Rora again changed its name to Aventis, and so the spin-off group were subsidized, sponsored by Aventis to set up their own business, which was one of the options for the redundancy. So I'm very thankful to a group of 4 scientists from Rome who set this company up, and then as soon as they were ready to expand the first time, invited me to join their group. And so I joined that group in 2002 as a principal biochemist within the biology group. And of course, I hadn't been doing biology for a large number of years. So I had to basically learn at speed, remind myself of screening and compound effects, and start managing expectations from the group there. We were all highly experienced scientists, so it's quite easy to slot in, and all the techniques I'd used in pathology were quite adaptable to biology. As we moved through the years, we had a number of name changes. So in 2010, we became Galapagos, and you'll see also had another name change towards the end of another period of time. But in 2003, the biology group were expanding and realised, because we had our own research as well, it'd be useful to do DMPK type studies, and so they set up an in vitro ADME group, and I was asked to join that group, especially as I wanted to go part-time. This sort of fitted in with personal circumstances as well. So I became senior scientist within the ADME group and helped set that up from scratch, basically. It was interesting, still fitted in with the type of pathology type work I've been doing before, still very disease orientated, and now I was looking at distribution of drugs and how they were metabolized within the body. And we were using isolated cells, hepatocytes, and isolated microsomal preparations to look at various drug metabolism enzymes, and we looked at plasma protein binding and permeability for blood-brain barriers. So all new techniques, and all had to be mastered in a relatively short period of time. But then as we moved on, I became a team leader in DMPK, and would have stayed in that field if circumstances hadn't changed. So by 2016, we'd already become member of Charles River and merged with a group which we were part of for Galapagos as well, which is Biofocus at Saffron Walden. So as part of the merger, they moved the drug metabolism group to to Saffron Walden, which left me with wondering whether I wanted a longer commute. So I applied for a position as group leader within biology at Harlow, where I was working, and I've carried on there. So moved back to biology, looked at various screens. Things of disciplines had changed by the time I was back in that group, but I'm certainly still learning and enjoying the type of work we do there. Very, very client focused, and large number of therapeutic areas and all sorts of different assays, still using some of the techniques I've used previously, but now having to pick up new techniques, but mainly leading other scientists at the bench and letting them learn the newer techniques. So in conclusion, I've been working as a bench scientist since 1987. And during this period, I have actually only worked in 3 buildings. I've managed to work with some of the same colleagues for over 30 years now. So some of my own colleagues are still working with me at Charles River. We have met plenty of new individuals from various areas of science on the way, and work with them and help them on their careers as well. Whilst I've only effectively been employed by 2 companies, it really is 6 different companies. So things have changed around me. I've not needed to look for new positions and new jobs. Been very fortunate in that respect. I've worked across 5 different disciplines in the time I've been working in the industry, and enjoyed a wide variety of therapeutic areas, explored working with a large number of scientists in the companies we work with as a CRO. And in that time, I've also managed 40 individuals, including IP students, PhD students, and at the moment, I have 5 apprentices, if we include the 3 master's students reporting into me. So very much engaged with Kent University as well. So I'm still learning new skills and still enjoying my time at the bench. And I hope that gives you some inspiration for, not worrying too much about the future, but enjoying the job that you're in at the moment.
Perfect. Thank you so much, Sharon, for this really lovely overview. And we really appreciate all the work you do, fantastic work and mentoring you do for level 7 research scientists. I think they are very lucky to have you, and we are very lucky to have you as well. So do we have, and we have time for one quick question to Sharon, if anybody would like to ask it now. Otherwise, we can use the chat as well to facilitate any question. But is there any question you would like to ask Sharon quickly now? One quick question. Anyone? No questions? Perfect. So if anybody would like to use...
Sorry, there is one in the chat, so I don't know if you want to read that out or leave that for Sharon to see. But yeah, there's one there from Casey. Do you want me to read it out?
Yes, I'm just actually looking at this. Thank you, Kirsty.
No
Yes. So Kate is asking, throughout your wide career, where were your biggest challenges and what piece of advice would you give us?
I think the biggest challenge is convincing people that whilst you were not necessarily an expert, because within biology we do have people that have remained in the same field, so they've remained as immunologists all the time, or oncologists, they come with a discipline and all of that's valued there. I think whilst I'm not an expert, I think you do not need to know everything in order to guide the work and make good judgments on it. You need to be able to probe and ask the right questions. So I think sometimes, certainly when I joined biology in 2016, I thought I'm really not experienced enough to do this role, but actually bringing different experiences into a discipline is quite refreshing, and I have definitely managed to carve out a role for myself within the biology group. So I think it's about having a little bit more confidence in yourself at times.